So just a little bit about myself. I'm an analytical chemist, which brings, one of the things I think about newborn screening, that's one of my favorite things about newborn screening. It's a multidisciplinary field. And someone like myself, I didn't know anything about newborn screening 20 years ago, but I had a good analytical background. And what I came to realize is that a lot of newborn screening is basically analytical chemistry of biology. And I always had an interest in biology, so this was like the perfect thing for me when I came into newborn screening almost 20 years ago. So the other thing that happened is on my very first day at work, I was told that the governor of the state wanted this certain condition, disease added to the panel, and that happened to be Krabbe disease. Maybe you're aware of it, maybe you're not, but it's a disease that had a lot of controversy, mostly because the therapy was kind of questionable, the test was new, and even how well the therapy worked was in question. And those are still some questions that are out there. So in that kind of light, though, it did give me experience with bringing on new methods and really trying to understand what it means to develop a screen and what does a good screen look like. This led us down the path, or led me down the path of working in a lot of other screening along the way. So I've had, I guess, opportunity and pleasure of being able to be on the forefront of screening for a couple other disorders. ALD is one of them, and Pompe disease is another. So before I get too much further, I want to talk about ScreenPlus. I do have a later slide that will repeat this. But what is ScreenPlus? It's considered a research study or pilot screening study, and the idea is to actually we use informed consent. We have coordinators in a hospital that are going to be meeting with new parents and talking with them, providing them information about diseases that we're going to be screening for that are in addition to the normal panel. Some of the pros and cons of doing that, so they have a perspective of the potential benefits and potential harms, and then they either sign up or they don't. And a lot of information can be obtained just in that part of the process. So that's a big part of ScreenPlus, is really just, and it has to do with how the family interactions and things go. Hopefully this thing works. Maybe I'll just do this.
It's not on. Sorry, it wasn't turned on.
So for those of you who don't know a whole lot about newborn screening, I'm just going to do a quick overview. It's internationally recognized as an essential public health program. It is something that the origins actually were in New York State with blood collection, and the Guthrie card. Rob Guthrie is the newborn screening, father of newborn screening. He developed a test for phenylketonuria, and that was out in the Buffalo area. So New York was involved with this, not so much as a state health program, but he took that and brought it to Massachusetts. Actually, it was the first state to implement. And New York was fairly shortly thereafter implemented screening for PKU. It includes screening of dry blood spots, also for screening for hearing defects, which happens in the hospital, and congenital heart defects, which happen in the hospital. Purpose is early identification of newborns at risk for conditions that are not clinically apparent at birth. So something like cystic fibrosis, sickle cell disease, these kind of diseases where a child looks completely healthy. That's some examples, and others are things like medium chain acylcarnitine deficiency, or MCAD, which is like a ticking time bomb for any, in the case where a child might be fasting for any reason, could then go into a crisis. So we're trying to pick these kind of diseases up that aren't apparent at birth and can be, so families and physicians know how to deal with it, which would aid in the diagnosis and treatment for these babies before there's any real bad problems that occur. So a little bit of overview of the process. It starts with the collection of the dry blood spot. This is ideally done between 24 and 36 hours. The blood card is allowed to dry for 3 hours. You see the Guthrie card up in the right and the heel stick. So that card, the blood collection and the filter, was the Guthrie, part of the Guthrie thing from way back in the early 60s. The sample was then shipped to the newborn screening, and there's real emphasis now on getting it to the lab as fast as possible. We want it the next day. So some places are getting same day or next day delivery. And this was something that's newer in the process, really the pushing for speed in the process because of some of the conditions having immediately life-threatening problems within less than a week of life. So the samples are tested for a number of conditions, and the conditions are generally on what's called the recommended uniform screening panel, and we'll talk a little more about that later. The screening lab will then, after screening, look at the data, evaluate for positive screens, and then if any positive screens are going to generally be retested, but there are occasions where if it's an emergency level that people will act on it right away and get a family in to see the physician so they can evaluate if the child has this condition because the clock is ticking. So the benefits each year, about 3.6 million babies are screened. And about a little over 12,000 are found with some serious condition. Accurate screening ensures effective babies are identified quickly, and there are no false positive negatives, so we don't miss anybody with one of these conditions. It's really important to not have a huge false positive rate, and that's always kind of a question, is what's an acceptable false positive rate, because anytime you pull a family in for evaluation, it's very traumatic for the family. Then it's important that, of course, the early treatment begins prior to the onset of the irreversible outcomes. This subpanel for New York State, most of these diseases are on the recommended uniform screening panel. I gave you some examples earlier, but congenital hypothyroidism, congenital adrenal hyperplasia, these are some of the more common disorders. Sickle cell disease, one of the more common, but then we have more recent things like spinal muscular atrophy, which is actually really common and has gene therapy, so it's one of the first diseases that made it to the screening panel with gene therapy. Then a whole list of mostly metabolic disorders and some lysosomal disorders that are on the panel as well. So there's a lot of biochemistry involved, biochemical pathways. Nearly all of these are from our genetic conditions, autosomal recessive, where there's going to be some enzyme block and markers. These kinds of compounds like phenylalanine would be elevated in the blood, or things along those lines. So in newborn screening, we're looking primarily for elevations of biochemical markers where enzymes deficient and that substrate or marker will be elevated in the blood. But with enzymes, we're looking for really low enzyme activity. And the ScreenPlus is doing we have a little of both of those going on in our panel. So one of the reasons, again, for ScreenPlus is, well, how do you evaluate what should be our recommended uniform screening panel? One of the key things is down in light blue, where it says requirement to nominate a condition. A newborn with a disorder must be detected and successfully treated through a prospective screening study. So this really puts a kind of what comes first, the chicken or the egg. You've got a disease, you've got a therapy, you maybe even have a test. And if you have a fairly rare disease, honestly, I feel this is a rule that's kind of like telling people they have to go get the witch's broom. Doesn't make a lot of sense to me, but it is the rule. So in one of the things with having pilot screening is to hopefully detect diseases in the process. Now, trouble really is the limiting factor is how many babies can you screen with a pilot study. So it's kind of not the perfect solution, and that problem still needs to go away as far as I'm concerned. But the process is a group would nominate the disease, which is an application. This takes quite a while. It takes a lot of experts. It's not a simple thing to nominate a condition. There's a review process, and the data that's put in the application is reviewed by a committee, and they decide, well, is there enough information here to send it to a thorough evidence review? And that would be the next step in the process. Go to evidence review, full committee, then would be given a report by that evidence review committee, and there would be a vote to see whether it be added to the panel The most recent condition, I'm proud to say, is Crab A disease. And might not be the best therapy, but the screen itself was so improved that it made it so there's really little harm. So you're going to detect people that have disease in this process. So if yes, the secretary signs, and then it goes on and ends up on the RUSP. So what's New York's prior experience? I told you about screening for Crab A, that was new, but one of the things that happened with Crab A, there was also other lysosomal disorders that were part of a similar screening mechanism that we could add, we could put all together and run, instead of just running for one blood spot and one disease, we could do what's called multiplex screening. And this allowed for running 5 other disorders at the time. And we had piloted that, we refined the process, we got it to, with the help of a lot of other people, Michael Gelb and Genzyme at the time, to help implement how do we screen for more of these lysosomal disorders, because there were enzyme replacement therapies available for all of them, or for the majority of them. And so we went ahead and we developed that method, and then that led to a project with Dr. Melissa Wasserstein, who was from Mount Sinai at the time. And she put in for a grant. We were funded through an R01 grant to do this pilot study, and that was called Lysosomal Storage Disorders Pilot Newborn Screen and Examination of Ethical, Legal, and Social Issues. And that went on from 2013, 27, sorry, I'm just on a block here, for about 5 years, and it was similar to ScreenPlus, where there was consent. The overall consent, people that uptake was 73%, but for those that had recruiters present or the hospital coordinators, it was 86% that signed up for this, and so it was a pretty successful project. Some of the lessons we learned is that we could do opt-in with consent. It was possible. Most parents did want to participate. One of the other lessons we learned is we had a fair number of false positives, and we tried to come up with ways of fixing that for our next ScreenPlus project we're working on now. And then post-screen, we did find that people, we were able to do this follow-up in natural history, and the individuals identified in that project are still being followed by Dr. Wasserstein and her team. So what we found out is the infrastructure worked. So I already told you about what ScreenPlus was. So this is the informed consent process. We got a bilingual coordinator that has direct conversation with parents. There's the verbal consent. There's also electronic materials for those that miss the coordinator, and it's with an iPad. They can sign up to be in this study. They learn about the potential harms and benefits, as I said, and then they get put into the REDCap database if they would like to be part of the study. Then what happens in New York, we wait until all the regular newborn screening is done, and then we'll match our birth records from the participating hospitals with the babies that are in this REDCap database that where the families accepted to be in the study, and then those children will be screened with the ScreenPlus assays. So the goals, again, I probably touched on this a bit, evaluate the analytical and clinical validity of our screening assays, evaluate the testing algorithms and determine cutoffs. We kind of determine the long-term impact of on the newborn screening and phenotype and outcomes. So anybody we're identifying, are we identifying individuals with a later onset, earlier onset? How do we equate the data from the screen, including multiple tier testing with what we're finding? What are the findings in clinical evaluation? And then, as I said, the ethical, legal, and social issues through, I'll show you some of the tools we're using to evaluate that, because that's really important. There's a lot of myths, potential myths and truths that it's hard to know about. What are the families thinking is the harms and benefits? Sometimes there's a lot of emphasis on this is a lot of harm. And sometimes there's not as much emphasis, this is a personal opinion, on the potential benefits. So Of course, newborn screening wants to minimize the harms, because we don't want to be in a situation where like we become the new anti-vax public health program. So that's something that we're really sensitive about. So here's the ScreenPlus panel. You'll see up here all the diseases. I'm not going to go through them, but some of these diseases, Fabry, Gaucher, are being screened by other states. Most of these, Niemann-Pick C, gangliosidosis GM1, and CLIN2, those lysosomal acid lipase deficiency, metachromatic leukodystrophy, these are all new examples. Metachromatic leukodystrophy is a disease that's likely to be submitted as the next RUS package, so it'll be up for a vote evaluation. And one of the things our data helps with is for that group to look at, well, what's the screening look like? Can a screening lab do this? And not just a private, lab in a college university, but can a public health lab do this kind of screening? And part of what New York does as well is help other states bring on the screening. So once we do it, we've got to go and we will bring other states in to help them learn. So the criteria is also listed here. Screening assay must be multiplexable, high throughput, reasonably priced. We have baseline validation studies. In other words, it's work somewhere. Generally, that's going to be a Washington lab. There's morbidity and mortality if untreated. We want a pediatric phenotype, and then there's got to be some kind of therapy. Those are all kind of normal rules of newborn screening. You need a test and you need therapy. Oops. So let me make sure I didn't miss anything. All right, there we go. So multi-tiered testing. So I was telling you earlier, when we first did our first project, it was all enzyme-based. We just looked at the enzyme assay. If it was low enzyme, we would refer. But what we've done since now is added second-tier molecular and biochemical testing. So the molecular is going to be generally Sanger sequencing, where we're sequencing the gene. And the biochemical, there's various markers, and like the primary markers for most of these would be the glycosaminoglycans, the GAG test, which we have done by Mayo, and that's going to be for the MPSs. So you can kind of do this. You can get a positive screen with an enzyme, low activity. Then what we would ordinarily do is we don't act on that. We're going to send it out for GAG testing or whatever other testing we need from Mayo. We're going to do we do in-house DNA, and then we evaluate those results. And that actually helps us not only with eliminating false positives, but also to figure out, well, where are our cutoffs? How are our cutoffs doing? So we start off purposely making them with a wide open window, and then we use these other tests to help us refine the way we analyze. And that becomes really important because most of the time in newborn screening with rare diseases, it's really difficult to get positive dried blood spot controls. So how do you know you got a good test? You don't know until you run it a while, and then you kind of say, well, you know it's going to be theoretically low enzyme, and then you're going to evaluate, well, what kind of molecular findings do we have, and what are the second-tier biochemical findings? And you use all this information together to help refine the test and determine where cutoffs in the algorithm and how it should work. So I'm going to skip now to some of the ethical, legal, and social implication studies. We'll come back to the testing stuff in a little bit, but I think this is particularly interesting. So we have kind of 3 levels of surveys we do. There's surveys on the consent process itself, parent surveys that are done after a month. So it gives some families time to not be, right after the baby's born, we have to put their thoughts together for us. And then also much later, like 6 months to 2 years. And there's things like, well, we want to learn about the consent process. Did they understand the information, why they participated or didn't participate? What are their opinions about expanded newborn screening programs and policy, dried blood spot research, which is a big thing nowadays. People are very concerned that that screening programs have their baby's DNA. And you can imagine the kind of tales that people will throw together as to the evil things we're doing with those samples, which is completely false. And we're very protective of those samples, and we don't give them to people for no reason at all. We're all in the interest of improving newborn screening. The other thing is, what do they feel about whole genome sequencing? That's a big thing now in newborn screening. Whole genome sequencing, you can sequence for any disease, essentially. It's just, what do the results mean? So there's trying to get a feel for what do families feel about that. We have a survey specifically for that. And then later, what do they think about, if they had a false positive, how did that affect them? If there was a baby's positive and they don't know what the outcome might be because there's uncertainty, how do they feel about that? But this is a really important part of this study. So some parental feedback, there's a lot of things, but here, a lot of reasons why they don't participate. The primary two is, one is they don't want their baby to participate in research, and another reason was that they feel like they don't need their baby to have more testing. That was sort of interesting to us, but we're finding out that we think a fair number get prenatal testing, and that families have some false sense of security that that's going to be all the screening that they need, and they don't need anything more. And that kind of screening is not perfect by any means, not that any screening is. So anyway, those are the main reasons. And I'm not sure if these slides are available, but if people wanted them, I'm happy to send them a copy. You can ask me for them. So the importance of the parental perspective, I did touch on this a bit. I think one of the things that we're trying to learn through these studies is what do the families, what's the public really think about newborn screening. And it might, we're doing the best we can. We have an ethicist and somebody who's an expert at developing these surveys. His name's Dr. Aaron Goldenberg. And we're trying to get very objective information about what the parents feel, so we can get information related to screening. There's a lot of information here. I don't want to get bogged down in it because I don't want to go over time. So potential benefits and harms of expanded screening. So the benefits are, direct medical benefit of the baby. That's the obvious one. These are some less thought of, but reproductive planning for parents. So if you've got a baby or a family that's found out they have a baby that might have a late onset disease and has a good chance for late onset disease, well, of course, that baby's going to turn older, and they may have another child in the meantime. So now they can do something with family planning to kind of prevent having another child that would be affected by one of these later onset diseases. Future planning for the affected individuals, so getting them the kind of care and help they need way ahead of when they would actually be detected. And this is probably the biggest one, avoiding a diagnostic odyssey for these families that have a later onset, or even any kind of, some of these diseases like Fabry disease, you're not going to develop disease until 3, 4, 5, 7 years old of age. So these kind of delayed, there's a long enough delay, similar to adrenal leukodystrophy, where you really want to be getting a head start on what you might be doing to stay ahead of this before the problem percolates and becomes worse for the child in the long run. Some of the harms, this is probably one of the biggest ones, the diagnostic and prognostic uncertainty. So you got somebody that might develop disease, and one of the things that's difficult is who's going to develop, who might develop. When you start doing screening, you find that there's a reason for a low enzyme activity, there's a reason for elevated markers in blood, but they don't always lead to disease. So how do we put all that information together? And that's why we feel this is more of a research study than just screening, because we don't want to put people into that unwillingly. It turns out that people do sign up for this, and with all our best intentions, some people end up, it doesn't really hit home until you're actually in the seat. And that's one of the reasons we have the studies to find out, well, what's the real impact to the families? So I spend an awful lot of time on this part of it because I think it's the part that's probably affects mostly what you as physicians and care providers for these families might have. And then now the other big thing is the privacy concerns, including data breaches and being unsure about how that data is handled. So we try to find out more about that. So now I'm going to come back to the assay development, pilot testing. It's really a team approach. As I said earlier, it takes a multidisciplinary team. So most of the work in newborn screening and developing tests comes out of one lab, which is sort of sad in a way. Dr. Michael Gell was the kind of the godfather of these newborn screening diseases for lysosomal disorder diseases by tandem mass spec. And him and I have been friends now for about as long as I've been at this, because he's the person who developed the method. Well, they still developed all the methods we're using for ScreenPlus, and they do a lot of de-identified pilot studies, and that's where you just run a bunch of blood spots where you don't know whose they are or what family they're from, but they're part of the use of residual dry blood spots that is important for us to develop the testing. So then the New York lab, well, we take what he's done, and we say, okay, can people with, there aren't PhD scientists working in a high-end lab, are we going to be able to take it and run it at high throughput means? And what kind of problems will we have? What are we going to run into for quality control materials? What's the long-term assay stability? Are there supply chain issues? Then we do cutoff refinements and things like this. And then we work with the CDC, they will work with us to help improve QC materials so that if this does go live, there's ways for people to be sure the test is working. Hate when that happens. Right. So a little more about the screening part. We use the research assays mostly based on biochemical pathways and enzyme deficiencies, as I said earlier. Developed a marker enzyme test using technologies such as tandem mass spec, immunoassays, and now we've got these more recent molecular-based assays. The beauty of those, again, is that once you develop one, it can work for many things. But the biochemical assays are thought to be still going to be more effective in the, not in the long run, but because they're actually seeing the function of the genetics. So if you have variants in a gene that you don't know what they mean, how are you going to, what are you going to do about that? And the biochemical assays are going to be are telling you, well, this gene really is producing an enzyme that's non-functional, for example. So we're going to determine that if the test is effectively differentiates between affected and unaffected newborns. And the challenge is lack of positive newborn controls, as I said, and the QC materials. So right now, with this ScreenPlus assay, we're really running an imperfect assay. We don't have the best controls, we don't have many positives, but we have, being a research study in the second tier and third tier methods, that's our tool to access how well is the test working. And all of this is going to hopefully lead to publications and data and information that will determine whether we should be adding these screening tests down the road. I'm not going to go into the details, but there are 2 assays, total of 3 punches to run 14 diseases. The rest is all chemistry details that most of you probably fall asleep on. We've set cutoffs for, these are the diseases, the top 1% for metachromatic leukodystrophy. We have cutoffs in nanomole, greater than 22 nanomoles for Niemann-Pick C, CTX greater than 0.11 micromoles, and all the enzyme cutoffs are somewhere between 10 and 25%, which we believe is fairly conservative in that we're going to throw a lot of samples at second tier testing. So scale up performance, we do make sure the assay works. We try to run as many known positives as we can, and we try to improve the quality control materials. Some of the quality control materials that exist now, we've tried to apply. It's kind of interesting. They aren't, so the CDC provides enzyme controls, and some of the enzymes are elevated in the controls that you think should be low. So we're having to deal with that. It's like somehow the enzyme survives their process of trying to kill it anyway, or make it denatured. We also look for scale-up issues. And then for any first-tier positives where we're running the DNA and biochemical, we're using all of that data to really evaluate our cutoffs all the time. That's the constant. We meet with the Mount Sinai, it's now Einstein College of Medicine, where Melissa Wasserstein is. We meet with them every other week to discuss our screening results and their follow-up results. So live screening, test babies from families that consented. We evaluate the factors that can affect the concentrations, for example, age of collection and birth weight. Screen positive newborns are referred for the diagnostic, and then we determine screen positive rates and all of that. This is all kind of normal screening process. Evaluate the needs and benefits of second and third tier testing. Any variants we find in the gene as part of the process, those are going to be kept in track of. If we start seeing multiple, what we find is if you see things often, but they're not, nobody's ever observed them before, variants of truly unknown significance, if you will, and those variants become cataloged, and we can begin to start to say, well, we think this is a benign variant. So if other people start screening, it's been seen enough that you can make some claim about that. Access to screen positive, assess the screen positive rate compared to disease incidence. So that's ideally what we would do at longer term. And then are there any effects from age and birth weight? Anybody that's positive goes for a clinical evaluation, physical exam, the diagnostic testing, and then we evaluate the diagnostic outcomes. The kind of outcomes are negative for the condition. That's an easy, it's easy if the diagnostic data says it's negative, the GAGs are normal, for example, then that's would be a false positive screen. If it's positive for condition, that's sort of a no-brainer too. Where we run into the hard work is what do we do with the ones that are in this kind of, we're not certain. So there are cases where you get uncertain clinical results and you got uncertain screening results. So these things, they go hand in hand. There's never a perfect line in the sand to determine. There's always a gray zone, and that's where a lot of information needs to be gleaned. So we tie it all together, as I pretty much said, compare first, second tier biochemical and molecular screen results. Got these easy cases, the not so easy cases. We evaluate the cutoffs and adjust them as necessary. Some ethical considerations. MBS functions within government, so all work must be accepted by the public, and that's part of a screening kind of rule from the very beginning. It's accepted by the public. So there's people think there's a main benefit for it, and they see that benefit. There's now more concern over testing with babies' DNA, so we're trying to assess that. And then identifying late onset diseases, and I alluded to this as well, babies' unclear diagnosis, what's the impact to the families? So all of this requires, though, this long-term follow-up, which is necessary to make some determinations. And that's why this is just will be an ongoing study long after the funding is done. So what have we found in this study? In the earlier years, 2017, we had 6 sites. Mass spec methods, we screened 65,000 babies in that time period. 7 Fabry's, 15 Gauchers, 1 Pompe. Pompe was added actually to the RUSP when we were in the process of doing the screening, and they did use some of our data for that. We had 86% opt-in rate overall. And Pompe, I already said that. And NPS was actually added to the panel as well in that time frame. So those 2 things went live, I won't say as a result of this pilot screening, but because of some of the help from this pilot screening. ScreenPlus 2021 to present, we have 8 hospitals. We've got molecular and enzyme and mass spec testing. This number is up to 20,000 now. This slide's a little bit late. We have 1 CTX case, 4 Fabry, 5 Gaucher. So we see a lot of Gaucher and Fabry's. Those are fairly common. Generally, a lot of those are more of the later onset cases, especially the Gauchers. Opt-in is 60%, so it's interesting. It's down. There's some reasons we think for that, but mostly the skepticism of 2020 and COVID. Is there any other things we can blame on COVID? So anyway, I feel like that's got to be the one, people are like, oh, government's got one, playing around, and I don't know, maybe gives a good feeling for what the negative effects of COVID on the public health system. So some of the biggest challenge, it does take 3 punches to do this. Ideally would like to do 1. We have 2 MS runs, so to do this testing, we have to use LC mass spec twice. We're trying to turn it into 1 test, and we think we're going to be able to do that. We really need positive controls. We can't get those. I've reached out all over the place. We can't get it, so we continue to kind of run blind with these assays. QC materials are still less than ideal. And it's tough to maintain these coordinator laboratory positions. Anytime you have a research job and it's got a 5-year time plan, people don't want to just work to the 5 years and then not have a job afterwards. So these, we're always kind of filling these positions, which kind of slows things down. And then there's lots of contracts. I'll show you in a minute all the people involved with this. So despite that, we know we can do large-scale newborn screening program testing, and they're necessary if you're really going to test the appropriateness of candidate conditions. We feel most parents are eager to participate. Fewer families participating in this study. I don't know, I guess I got my numbers mixed up, 73 versus 86. I think it was 86 before. But we think possible more prenatal genetic testing is one of the reasons, then the increased privacy concerns, as I mentioned. Informed consent models can thrive despite some external stressors, because we were able to do a lot of this actually during COVID. Then ScreenPlus provides an example of what we think is a successful, mutually beneficial partnerships between academia, government, industry, and patient advocacy groups, which are all involved, and you'll see this slide. So this is my last and final slide. You can see it does really take a lot to run these programs. The Einstein College of Medicine is where the headquarters, and they're the primary principal investigator, Dr. Wasserstein and her crew. Myself and my crew, somebody who's not mentioned here, Monica Martin, who developed a lot of these tests in my lab, and Gelb's team, who are developing the assays. Then there's, we have pilot site principal investigators that are going to receive some of these cases and have to do follow-up. Finally, we do have advisory panels here, and you can see all of the industry partners. All of those partners have contributed money, so this is a government-funded and private industry-funded project. So that's my talk. I thank you for your attention.